one tag that says review for final. There's going to be another tag that says essay questions for final. Okay? All right. For those of y'all that got in here later, I do have your grades and what you need to make on the final to make either a C, B, or an A. So get with me after class, I'll show you that. But otherwise, first of all, I did not receive not a question from any of the general biology classes. At all. Period. None. So, you're getting my test. Should be fun. But I decided to take it a little easier on y'all. And what I did was I just took questions directly from test 1, test 2, and test 4. Okay? So it's pretty much the same material. So, I'm going to go through. You might want to grab a pen. Start taking notes. I'm going to run through this fairly quickly. Oh, and like I said, I should have, my goal is, as I'm shooting for, having all the evolution projects graded by Friday and having them up by Blackboard by then. What's going to end up happening, since I found a large group of people not showing up to other people's presentations, basically I've been keeping track on those cards you've been handing in. So if you didn't show up, I'll give you one mulligan, but if you didn't show up to several of the other presentations, you'll either lose your +5 bonus for presenting, or you will be docked 5 points. So, all right, first concept you need to know about is endo and exocytosis. Essentially know what processes use it, and then a description of the process. Know what the function of a cellular junction is. Reverting back, I believe it was chapter 2 that we did all the chemistry on. Do you remember the Bohr models and finding valence shells on the electrons? You'll need to be able to do that one. Okay, next concept is cell motility. You'll need to know pretty much what causes it in the cell, and what's structures, and also what cells in your body cause movement, i.e. muscle cells and what they're constructed out of. Okay, know the processes for breaking down using enzymes to break down unused organelles in the cell, or also, cellular digestion. That's pretty much the lysosomes, the peroxisomes, that kind of thing. Know the different levels of complexity inside a living organism. You know how we started out at atoms, we worked our way up to molecules, molecules then become cells, cells become tissues, and then tissues become organs, organs become organ systems, and then organ systems become organisms. Know the different classification levels for, say, species, populations, communities, that kind of thing. Again, we'll probably hit upon this a couple times, but know the processes of cellular respiration and photosynthesis. Understand both where the process is happening and what the process is. Know the different types of microscopes, like the compound microscope, the electron microscope. Remember that little figure that we looked at that tells the, obviously tell I'm not an English major. Remember the figure that told you to what size the microscope could look down to. Okay, like you start out with using the light microscope, and then you use the phosphorescent microscope to look at specific proteins, then you look at with the electron microscope to some stuff smaller. And then understand transmission and scanning electron. Know what glycoaminoglycans and proteoglycans are and where they're found. And that's essentially connective tissue, cartilage. Okay, know what an amphipathic molecule is? Be able to use the terms catabolic and anabolic and understand what they are. Understand the first and second law of thermodynamics. First law is that energy can't be created or destroyed. Second law is that any change in energy from, say, mechanical to electrical or electrical to mechanical, you lose a small percentage of energy because it's increasing the entropy in the universe. Know how to use delta G, that change in energy, and understand whether a negative or a positive is equal to, say, a catabolic or anabolic reaction. Or also understand which one's an endergonic or exergonic reaction. And if you remember, one of the specific questions we had on here was the delta G use of ATP. So be sure to know how to use that one. Okay, know how biochemical regulation goes through and actually overcomes a co-inhibitor. Understand what would happen if you altered the three-dimensional structure of an enzyme. Oh, and secondarily on top of that for enzyme, understand, how should I put this? Understand enzymes are used in a very specific range. Most of them are judged by temperature and pH. Okay, do y'all remember the process that we use to replicate DNA for DNA fingerprinting? No, the process actually doubles it. PCR. Understand the difference between a genomic library and one of those written like this, a cDNA library. What's the difference between the two of those? Oh, these last few questions y'all should be hitting on because they were test board. Okay, understand gene amplification. Understand what it takes to make cancer. Okay, figure out which type of mutation can actually change a proto-oncogene to an oncogene.
What to what?
Convert a proto-oncogene to an oncogene. Okay, I want you to also know okay, the MYC gene. Know how it's associated with cancer. It's spelled MYC, and it's usually italicized. Know the process for the dideoxy method for DNA sequencing. That's that one sequence where they go through and they add certain base pairs that where if they're added to the DNA, it stops it. And in that way you can look and it actually sequences the DNA in order for each base pair. Know the purpose of a selectable marker whenever it's associated with an antibiotic resistant gene. Know the different types of tumors and what they're called, depending on their location in the body. Know what ionizing radiation can cause. Okay, know the different types of repair enzymes for DNA. If you didn't read it before, read about ectodermata pigmentosum. And then know what it would be to be a clone on an intelligence level. If they give you examples, you need to be able to tell which one's the clone. And that's it. That's the 50 questions.